like do the motions and the techniques necessary to do the tissue culture following a kit, but also read the history at the same time, and eventually the theory and the practice coalesce through, a fuck ton of failure. But yeah, like at one point I thought I was going to be a mycologist because I was just so good at growing contamination. Like I tried to do tissue culture and I would just get fungus everywhere, and I'm just like, fuck it, maybe fungi is cool, right? Like I always try to find the
fungi.
bright side. Yeah, sure. Yeah, I just wanted to roll with that joke to the logical extreme.
Hi everybody, welcome back to another installment. So today we're going to be highlighting another key member of the Biohacking and Genetic Engineering Design Group. Today we have Sebastian, which is a moderator of the group. He's been a long-term member, a long-time biohacker, and I'm super excited to interview him for this portion. So, hi Sebastian!
Hi Esther, how you doing?
Yeah, he's a little bit under the weather still because he actually caught COVID, and he's kind of alive, but mostly dead. Not talking about his soul; he's already dead inside there.
Yeah, dark.
That's my type of meat, you know that. So yeah, I would just love to, ask him a few questions about how he got started into biohacking and what kind of projects he does and how he greatly influences the community in his great ways. So yeah, let's get cracking. Cool. So yeah, Sebastian, do you mind just telling us who you are and how you got started in the biohacking space?
Okay, where to begin? So I'm a human being in New York City, and born and raised here, and started kind of interfacing with the biological world as more of like staring at leaves, cutting up my mom's flowers from home. Just really curious about the plant as a material, because I couldn't really parse the idea of it being alive, but stationary at the same time. Right, so it was like on a different time scale, and this was like me being around 8 years old, where I was like starting to get understandings of like life sciences, but like Babby's first lesson, like here's a cell. And a lot of those textbooks showed some images, a lot of it was hand drawn, and I never really got a connection to it until one day my aunt, just trying to figure out how to get me into schoolwork, because I've always been a really crappy student. I'm really interested in science, but just a terrible student. So my aunt, trying to figure that out, got me a microscope kit, like a really, really cheap, bare bones, barely works microscope kit. And I loaded it up, I threw the manual out the window, and the first thing I did was I put a slide under a microscope, and I just like rammed the objectives through and broke it. Like I had no idea what I was doing, right? And after like maybe a week of just like breaking slides and just not knowing what I was doing, my dad was just like, what actually are you doing? I'm like, I want to see the thing. I took a pedal and I put it underneath and I just wanted to see it. He's like, Jesus Christ. Okay, here, sit down. Let me show you how to mount a slide, right? So my dad's a physician, but he's classically trained from like way back. He's like born in the 50s. So did a lot of gram staining by hand and all sorts of microscopy as part of his training. So he actually really liked that stuff. So he wanted to show me just some bare stuff. And he took some pen ink. He's a big fan of this like one nice like cheap workman's fountain pen that has crystal violet as its ink. It's a French student ink, and basically that's also a microscopy stain. I had no idea at the time, but he added a little bit to a piece of tissue, put it under the microscope, and he's like, all right, go take a peek. And I looked at it and I'm just like, oh, holy crap, that's the thing from the textbook. Right, those are the little squares, the cells. So I'm just like, okay, life has cells, confirmed, experimentally verified that this is a claim, right? I had no idea that part of science was so important, right? But it's more of like foreshadowing, like realizing that seeing it for myself is the only way to believe it, because being taught and like lectured to just didn't really work. So fast forward a couple years, and I started just like expanding on how to interface with the natural world in some capacity. And I just found plants to be super cheap. And my mom's got a green thumb, so there's plants everywhere. So I had like plenty of victims to sample from. And I don't like working with things that scream when I cut it. So I stuck to plants, like mammals, not my thing. So that being said, eventually I found out about tissue culture. Right, and cloning for me was a big buzzword. I'm just like, whoa, what's cloning? And so like, long story short, I started learning tissue culture from from forums, eventually got into orchids because my mom loved orchids, started flipping orchids, like taking orchids from the trash, putting them under lights and hormones, got them to flower, sold it back, made a profit. Advanced my technique until I stumbled upon genetic engineering in college at Stony Brook. And I didn't want to do HIV research or cancer research, so I had to do a lab in order for me to graduate. And so I worked in the only plant lab there, and it happened to be a Vitaly Sitovsky's lab, which is the one of the grandfathers of agrobacterium, the main bacteria we use to transfer DNA into plants. So I indirectly had one of the best opportunities to study and understand plant engineering, right, from a formal perspective. And I mean, I was trash. Like I still no talent. This is pure just like grit and perseverance. Like just miserable with note taking and all that stuff. But that stuff stuck with me. And that initial experience of just like looking through a microscope and verifying the stuff that I was being taught kind of like became a theme for my life. Like I really wanted to do everything I was learning in that plant engineering lab at home. So I slowly started cobbling the tools together and mimicking those processes at home. And because the United States has this interesting gray area when it comes to biosafety level 1 and agrobacterium transformations. So as long as whatever I'm doing doesn't cross the state lines without a permit, there's no law that says I can or can't do what I'm doing, right? So basically just followed that to the logical extreme. And now I offer my services for genetic engineering as a for-profit company. And then utilize those profits and funnel it into my nonprofit, where I just distribute all the stuff that I've built so that I can get more people to do plant engineering, basically.
That's awesome, Sebastian. Yeah, you have a very interesting background of not necessarily traditional academic, but you're also very self-motivated to start projects yourself and take them to term. And plus, there's not so many plant engineer, like bioengineers in the world, because it just takes so fucking long to grow a plant.
Yeah.
But like, yeah, can you take us through the process of like, how do you self-start a project from literally nothing? Like you said, you had, you found some resources online. Like what takes you from A to B when it comes to new projects?
Okay, so there's kind of two answers for me. One is the way I tried, which is hit your heads against the wall for 15 years and try to like get things to work. And then what I learned in hindsight is break your interest into a single question if possible, right? It could be broad, right? Try not to be like lofty, like I want to cure cancer, right? Like just focus, and through that focus, build your lab around that project, right? And when you do so, you're also not just building the resources of your lab, you're building the wetware and thoughtware, right? So wetware being like reagents and living material, but then the thoughtware is the most important. That's the actual literature review. That's the things that gets you to that point, right? So what I've learned in hindsight is that before starting any project, and this sounds obvious, but like for me, I sucked. What the lesson I learned was that you always start with literature review. Figure out what's been done before your time. And the further back in time you go, the more accessible the experiments become, because they don't rely on super fancy equipment or supercomputers or just a tremendous amount of prerequisite knowledge for you to even start parsing that journal, right? So like when I started getting more technical into tissue culture, I started reading the history of plant tissue culture, and not like one book, but literally like going on Google Scholar and just taking the range of the dates back to like the 20s. With like Wendt, or like this 1962 with Morashiga and Scooge, those like seminal papers that defined what tissue culture was. And so like versing myself in that while failing, I found to be super important. Like do the motions and the techniques necessary to do the tissue culture following a kit, but also read the history at the same time. And eventually the theory and the practice coalesce through, a fuck ton of failure. But yeah, like at one point I thought I was going to be a mycologist because I was just so good at growing contamination. Like I tried to do tissue culture and I would just get fungus everywhere, and I'm just like, fuck it, maybe fungi is cool, right? Like I always try to find the bright side. Yeah, sure. Yeah, I just wanted to roll with that joke to the logical extreme. But yeah, eventually I started getting better by just like trying a bunch of things, and then the first thing that worked, like looking back at my notes and praying that I wrote it down. Right. So the second most important thing is, holy fuck, write it down. Write everything you do down. Keep it like in a journal. Like don't even think about lab notebooks as this like super fancy, organized, structured thing. Just to like imagine a science diary. Today I did this stuff. Throw in some personal notes, whatever. Just be able to write in a tone that's writing for your future self that forgot all of this shit. right? Because in 6 months, if you for some reason put your pipette down and stop doing stuff, I guarantee you'll forget what you've done, right? No matter how good you are, you're going to get rusty. It's like a language that you work with. So, or like playing a musical instrument, you'll pick it back up, but like having a reference to look back on and seeing how your mind's progressed through just like handwritten or hand typed lab notes has been such a benefit for me. So, I mean, now I'm quote unquote eloquent enough with biology that I can come up with some like slightly more creative ways to implement projects, right? Like I have an idea in the shower, just like, hey, I just want to try candy cane, sugar cane, right? Like I have this circuit that turns any plant just blood red. And if you put on a transposon, it'll activate only in certain cells, and the patterning could result in like a spiral shape. So you can have a sugarcane plant that actually has a candy cane spiral, like a red spiral, or like cool patterns, right?
Yeah.
Because for me, the loftier goal is to be a flower designer one day, right? And turn like things like these into flowers that no one's ever seen before, right? Kind of like Willy Wonka minus the exclusive bullshit and strange like little slave labor people. It's like open source Willy Wonka where everybody can do this, right? Because I find a lot of design philosophy and a lot of design people to have like massive egos. No offense to the folks who are like, in the trenches of design, but like when it comes to like artistic expression in biology, it tends to be super highfalutin, and I just don't jive with that. So part of my practice is to actually open source this stuff. As I learn something, as soon as it works, it becomes an open part. And I'll either send it to you for the cost of shipping, but if you can afford it, I'll sell it to you for like $10. Like I don't want to make cost a barrier. Just justify the loss of reagents to deliver it to you, that kind of stuff.
And, Sebastian is definitely one of those people where he has his entire lab book online on Facebook for you to just like browse all of the data. And you can see behind him, he's actually not in a lab. He's not in an official lab, but this is his own home lab that he's built over time. So take us a little spin, C's.
Okay. All right. So let's start there. So here's my plant rack with all the plant stuff. Behind me is some devices that I made, a tube tumbler so I can replace this thing, which is a shaker that costs like $700, and this thing cost me like $6 in parts, 3D printed with a motor, and it's dead quiet. It does the same rotational thing, but just this way at an angle. Then some more hardware, like here's a shaker incubator. Here's, sorry for the shakiness, here's you guys. There's just some, what should we call it? Pipettes. Super stupidly fancy PCR machine that I can't believe I have and I saved up so long for. The heart of my lab, which is this laminar flow hood. There's an Opentrons that somehow fell off a truck. There's a nano drop spectrometer over there that I got for like $200 with a blown fuse. And then that thing over here is my gene gun, which allows you to shoot DNA like a shotgun into whatever tissue you want. And I might be the only one on the Eastern Seaboard that has this model.
Yeah, thanks for the lab tour, Sebastian. He is really living proof of what you can do in terms of like bringing science to your home and conducting experiments, on a very basic to even an advanced scale. And I think a lot of the community just look up to you as like, a model of like what you can actually be and what kind of science you can be doing on like a very impactful level. So Sebastian, I would like to just kind of take this time right now to find out how we can get involved into building this mega lab like you, and to emulate kind of like the lab work that you've been doing. So what kind of groups can you tell us about?
Okay, so I guess the first and most important point is that you don't need a fancy lab to do good science. What I'm starting to realize is that all the fancy stuff I have has like, fate's kind of dealt me a funny hand where I managed to flip equipment and afford all the fancier equipment by like fixing broken stuff, selling it on eBay, taking the profit, saving it, whatever. But I started to realize that a lot of these equipment can be simplified. And there's so many people in the formal academic communities in biology, especially producing tools for affordable labs in, let's say, field labs or in hospitals in rural areas or things like that. And a lot of medical adjacent technology is molecular technology, right? So one of the initiatives that I started is called the Pedalsmiths Plant Engineering Research Party, which right now just resides as a Facebook group. It'll soon just be a forum on a proper web page with, like archival posts and stuff. Less immediate feedback, but it's archival and searchable. And the goal there is to try to grow plant engineers from scratch, right? So just out of just kind of feeling a little bit, for lack of better words, just a little bit lonely in doing this practice and realizing that it's actually not that difficult to do all of these things. And there's a lot of equipment fetishism going around that makes it inhibitory for people, thinking that you need a $5,000 PCR machine to do PCR, where like that artist Paul Vanus did it with 3 pots of water near a campfire, right? So like breaking down the problem into its fundamentals makes you realize that you don't actually need all this stuff. So so in that spirit, I basically, over the holidays, I sent a small test group, 13 of my basically workhorse plasmids, and then like slowly growing them up so that they can actually do biology with plants, where like I'm not going to dictate whether it's commercial or whatever. The only thing I ask is that you share alike, right? So a lot of the stuff I do is licensed, but licensed in Creative Commons. So as long as you share alike and you maintain that open sourceness, you could do literally whatever you want with my stuff. Like rip me off, just give credit where credit's due. So yeah, in that Facebook group, basically, I'm just trying to mentor a really small, tight-knit community of people from all sorts of academic and non-academic backgrounds, and just try to like grow a community around the research culture of plant engineering, if that makes sense. Because one of the key important aspects of growing a community is to develop a kind of research culture where like people have internal self-motivated incentives to produce research for the community at large. Like not just I want to make a glowing plant or I want to make a plant that's blue, but have like something more structured of a question, right? Something that could actually generate understanding that can help other people. And one of the first things that we're doing is working on tools and techniques, right? So for the next year, I'm basically dedicating myself to getting as many people up to speed with the techniques into a level where people can teach those techniques to others, such that they don't rely on like me giving a lecture, right? So that type of one-on-one mentorship is really difficult to do in massive scales, but on small scales, if you can teach people enough so that they can teach others, it spreads like a virus. So I'm basically trying to make a plant-demic of sorts, where it spreads, but it's information, not terrible diseases.
How appropriate for this time. Very dark as well. Yeah, so join Plants Pedalsmith's Plant Engineering Research Party. It'll be in the links down below, so you can get access to it. And I know, Sebastian, you have a Patreon, I believe.
Yes, yeah. We've been working with ways in which we can produce content that's not necessarily video specific, just because that literally is a full-time gig to do videos nonstop, and because we take no profit in any of the stuff we do. It's like literally a non-profit in the purest form. Everything goes into lab overhead, and I've been so busy trying to set up the actual like wet lab tools to distribute that like producing content was very difficult. So for the first 2 years, it was kind of like touch and go with the production of what we're trying to do. And now we're slowly starting to realize that for the small amount of folks that are interested in actual biological research, that's going to be basically for all of our research drafts, our manuscripts. Like we're about to submit to bioRxiv and then hopefully in PLOS sometime this year. One of our papers we've been working on for the last like 2.5, almost 3 years. And it's like a 40-page massive document of what we've done. And chiseling through that is complex and difficult because like a lot of it's from the heart because we're speaking as amateurs, right? And you, as a Patreon, you would get one of the early access to those drafts. You could submit your own criticisms. And just basically honing in on just folks that want to do biological research, like pure classical biological research. And I mean, of course, if you'd like to donate, for sure. But yeah, that one's still in the works in terms of like how to actually interface and produce content that's worth the monthly subscription.
Okay, and I heard you started an Etsy store.
Yeah, one of the few people that aren't reselling plasmids. I'm actually selling my own handmade plasmids on Etsy, as well as seeds for tobacco, which is my favorite model plant of all time because it's so easy to work with. I also have this wonderful melanin plasmid. That's the melanin plasmid. That's a negative control. It produces melanin and it's optimized for plants. So you can, I haven't tested it yet, but essentially I'm working towards making jet black plants, like obsidian petal plants. And that's going to be on the store, and everything that I'll be selling is open source. So I've been working a lot on trying to cobble the bits together to avoid patents and kind of make everything into a homogeneous, standardized ecosystem of parts that you can play around with. And they're always going to be like 10 or 20 bucks.
So go check out his Etsy store. Everything is homemade and handcrafted.
Yeah, bespoke with love in Long Island City.
That's awesome. Okay. So we covered that you don't have to have a fancy lab like Sebastian's to get started in plant engineering. And what we really want to hone in on is just learning those, small basic tools and techniques to teach to other people to get involved with a larger picture of hopefully creating your dream of genetically engineering plants in your fantastical, magical garden one day.
Yeah.
So thanks so much, Sebastian. That's kind of all of our time. And yeah, if you'd like to get in touch with me to, do one of these like interviews, please shoot me an email. We have, I'll be releasing these videos once a week. And please join our Facebook group. We have lots of activity going on right now. And also, if you're interested in buying me a beer or a coffee, feel free to donate to and support this channel, because I have a couple of people that are working on this project with me that we're creatively collaborating on to give you some of the best content and to interview some of these fantastic people. So thanks so much for your time, and be well.
You too. Bye.
Bye.